Analysis Match enables you to automatically discover other analyses that have similar or dissimilar biological activities compared both to your already run analyses or to tens of thousands of analyses curated from publicly available databases and stored in IPA. Each row in the table represents a different analysis. It may either be one of your own or one of thousands of curated analyses. In this example of gemfibrozil-treated rats, I've filtered to find the most significant results. Gemfibrozil is a PPAR alpha agonist, so it is not surprising that the top scoring match related to my analysis is from a study that used another PPAR alpha agonist treatment. This result confirms the biology I am seeing. Now, I'm going to show you how I was able to filter down to these results. This is what the analysis match results look like when you first select the tab. By default, the rows are sorted so that those most closely matching the analysis with which you started are at the top of the table. The Project button allows you to filter the matches based on whether the analyses were performed by you or are from the publicly available OmicSoft curated datasets. By default, the columns describing the matches contain metadata such as the disease state, the name of any gene that was targeted in a study, the type of tissue used in the experiment, any treatments, the comparison, for example, a treatment versus a control, and more specifics of the type of comparison, such as the exact treatment. If you would like to view information about the matched analysis in OmicSoft's Land Explorer, you can click on this hyperlink. We also include a hyperlink to the source of the data. You can add more metadata columns to this table by clicking on the Customize Table button. For example, you can filter the matches by gender, cell type, and more. You can also evaluate the metadata for your matches by selecting them in the table and then choosing the Evaluate Metadata button. This allows you to view which metadata terms are significantly enriched. The remaining columns provide information about the matching z-score percentages, which are used to quantify similarities and differences between matches. IPA matches canonical pathways, upstream regulators, causal networks, also known as master regulators, and downstream effects, in other words, diseases and functions. You can also view the p-value scores for the same entities by selecting these options in Customize Table. As mentioned earlier, by default, the results are ranked based on the overall z-score, which is the average of the z-score percentages for all entities. For this video, I filtered by the overall absolute z-score to find the most significant matches. The z-score percentages run from 100% positive to 100% negative. We chose this scale because it can be powerful to see not just what analyses are like yours, but also find ones that show very different results. Positive z-score percentages indicate that the matches are similar and are shaded in fuchsia, while the analyses that are different have negative z-score percentages and are shown in light blue. You can think of them as anti-matches. You can learn more about what is driving the matches by selecting the analyses of interest and then clicking on the View as Heat Map button. This new window provides you with a heat map, with each analysis shown in a column, and the name of your analysis is highlighted in pink. Each row represents the name of an entity. The dendrogram on the left side of the screen shows what entities cluster together. You can choose the Cluster Columns option to view the heat map as a function of clustering similar analyses together. Clicking on the header of an analysis shows its metadata information. To view more details about the matching analyses, you can select the View Comparison button to view the analyses in a comparison analysis. You can also select this option in the Analysis Match table. For more information about analysis match results, you can go to the Help menu to access our online help manual or watch our video tutorials and webinars.